Alzheimer's is one of the world's cruelest diseases, robbing millions of people of their health and quality of life before ultimately taking it. Women and people of color are hit hardest. 2/3 of people with Alzheimer's are women. 40% of Alzheimer's patients in the U.S. will be Latino or Black by 2030. Tough problems demand innovative solutions. That's where Us Against Alzheimer's leads the pack, focusing on preventing the disease, detecting it early, and making sure everyone has access to treatments. On prevention, we built a national coalition to persuade the federal government to make Alzheimer's risk reduction a national priority. Our Brain Health Academy educates healthcare and wellness professionals in communities across the country. And our campaign, in coordination with the Centers for Disease Control and Prevention, reached more than 1 million Latinos and Black Americans in 2022 with culturally tailored information about Alzheimer's and brain health. Early detection and diagnosis are incredibly important, increasing the possibility of effective treatment while enabling people to make plans and spend time with their loved ones. Brain Guide has given more than 500,000 people the opportunity to complete a memory questionnaire and get resources tailored to their results. And we all want qualified patients to have access to safe and effective treatments. Us Against Alzheimer's is pushing for clinical trials to include more people of color, seeking accelerated federal review of treatments, and calling for Medicare to stop treating Alzheimer's differently than other fatal diseases. We're not in this alone. Our work is guided by the voices of patients and caregivers as we join on a shared cause alongside a broad coalition of scientists, policymakers, industry, and advocates. Together, we're bringing help and hope to the people who need it most. That's the power of us.
There have been major developments over the last year when it comes to developing treatments for people living with Alzheimer's, the focus of our first panel of our 2022 National Alzheimer's Summit. With multiple treatments moving through the pipeline, there's good reason for patients and caregivers to have hope. Our panel of researchers and government leaders will discuss the state of treatments today and the potential and promise for the months ahead. Here's the moderator of our first panel, Debra Lappin, of Us Against Alzheimer's.
Hello, and welcome again to the Power of Us, the Alzheimer's National Virtual Summit, and our opening panel, Drugs in the Pipeline, What Next, and the Science Behind It. We could not have a better panel to explore this important opening topic. Dr. Eliezer Masliah, Director of the Division of Neuroscience at the National Institutes on Aging. He has remained and remains today in an unparalleled position to look at our nation's public sector, $3.7 billion, as George just told us, in Alzheimer's and the promise it holds. Dr. Dennis Selkoe, co-director of the Ann Romney Center at Harvard for Neurological Diseases. But more importantly, Dr. Selkoe is probably the most cited author in the world on the role of beta amyloid in Alzheimer's and the underlying molecular basis that the promise of this science holds. And Dr. Yaning Wang. And Dr. Wang is now in the private sector as CEO of Createrna, but before that, he was in the room. He was a director of the Division of Pharmacometrics in the Office of Clinical Pharmacology at the FDA. He was there to watch as the drama unfolded around Adukanamab, a drama that we'll discuss today. We look forward to our time with each of you. Let's begin. Dr. Selkoe, there are 4 current drugs that we know in the pipeline that are known as anti-amyloid monoclonal antibodies, MABs. You know these drugs well. What is an anti-amyloid monoclonal antibody? What is the amyloid cascade? And why have you invested so much of your incredible intellectual capital in the belief that this approach holds such promise for people with Alzheimer's?
Thank you, Deborah. I'm delighted to be with all of you. An anti-amyloid antibody is a natural molecule, an antibody, like we have antibodies to COVID and to many other infectious diseases, but it's made against this key protein that accumulates in the brain, so-called amyloid beta protein that makes up plaques. So an anti-amyloid antibody is an antibody very specific for the amyloid protein. It's sort of like a key and a lock. So the key is the amyloid antibody, the lock is the amyloid material in the brain. And the goal is to have that antibody touch and bind to and remove the amyloid. The amyloid hypothesis has been around for a long time, virtually since Alois Alzheimer described this terrible disease in 1906. And it's been the idea that a peculiar substance in myriad plaques in the brain might be important for the beginning of Alzheimer's, might even be causative in some cases. And there's been a lot of evidence that at least familial forms of Alzheimer's can actually be caused by amyloid buildup, because there are mutations in the amyloid parent protein, which we call APP in our field, and the enzyme that cuts it, an enzyme called gamma secretase. Beyond that, conventional patients with Alzheimer's disease, patients with late onset typical Alzheimer's, also have a buildup of amyloid beta protein in the brain, just like familial forms of the disease. And so the hypothesis was that if we could remove that, it would not be likely to trigger downstream events. And many of us in the field believe the most important downstream event is the buildup of the tau protein, a brain neuronal protein that builds up as tangles. So the amyloid hypothesis, in short, connects the two main lesions that Alzheimer described back in 1906, the amyloid plaques and the neurofibrillary tangles, and suggests that the amyloid material is upstream or comes just before the tangles build up. And so we're very excited to talk about these 4 antibodies today that have the ability to clear amyloid from the brain, and therefore could well be what we call disease modifying, not just symptomatic treatment, but actually modify the underlying brain pathology.
So let me ask, you just said there are 4 antibodies within this class.
Mm-hmm.
And just so we have a common footing as we go forward, just give us the names of each of these MABs, because we may start referring to them, and their order in the pipeline, because this is about the pipeline.
Absolutely. So the first in the pipeline of 4 is Adukanamab, which received the brand name Aduhelm from its creator, Biogen. So Aduhelm, as we'll be discussing today, was already given accelerated approval by the FDA on January 7th, 2021. The next in the pipeline is called Lacanumab. They all end with MAB, standing for monoclonal antibody. So Mechanimab comes next, and we've heard very exciting top-line results on September 27th that we can speak about today. And then the next to be declared in terms of its trial outcome will be Gantenerumab. And finally, probably in March or so, the 4th antibody in the pipeline, Donanemab, will also have its results revealed.
Oh, that's perfect. Let's stop there. We will be coming back for much more, but thank you for your clarity as our mentor in all of this. Dr. Masliah, let me ask you, you sit, as I said, with $3.7 billion that NIA now has to invest in finding new, understanding the basic biology and translational research. What has been the NIA's role in bringing these monoclonal antibodies forward into development? And what excites you about this class of therapies?
Thank you, Debra, and thank you for inviting us to be here today, and for all of you for joining us on this exciting panel. Well, yeah, I mean, of course, Aduhelm and Lacanumab and the other monoclonal antibodies that Dr. Selkoe mentioned, of course, have been developed with industry funding. Nonetheless, I think that NIH and NIA have paved the way for many of these clinical trials, in particular, as we have been instrumental in funding, for example, many of the ideas about the amyloid hypothesis that Dr. Selkoe mentioned throughout the years, as well as, and very importantly for clinical trials, the development of biomarkers. For many years, when we were initially testing some of these monoclonal antibodies in Alzheimer patients, we didn't have biomarkers that would tell us if these monoclonal antibodies were changing the levels of amyloid in the brain or not. And it was really very difficult to select the patient population, to select who was responding or not. So the development of biomarkers, again, with federal funding, has been quite instrumental in now the current success with the new clinical trials with anti-amyloid antibodies. Furthermore, there have been many other outcome measures that have been developed thanks to federal funding, including clinical measurements that are needed for these studies, genotyping study, meaning understanding what genetic background individuals included in these trials have, and also to the imaging studies, the brain scan studies that are necessary to see if there are any complications deriving from the treatment with these monoclonal antibodies. And we can go into a lot more detail on all of these, but I think the community have come together to the challenge and have done a really spectacular job coming up with new projects, new ideas, and moving forward the field to where we are right now. And this is, of course, thanks to the working together of industry, with academia, with the government, the federal sector. And we're really proud about these public-private partnerships to advance research on Alzheimer's disease.
Thank you for that. I know Us Against Alzheimer's believes so strongly in the power of the public-private partnership, and you all have been leading those. Talk to us, you mentioned biomarkers, and let's stay with our monoclonal antibodies. What is the biomarker that has been used so that we know first that people going into trials have amyloid that is delivered by pathology of Alzheimer's, and secondly, that you knew that the drugs we're talking about were effective in removing that amyloid burden? What was the biomarker?
Yeah, essentially, there are three of them. The main one is essentially a brain scan where an injection is given with a chemical that actually binds to the amyloid plaques in the brain and basically makes them, let's say, shine or be apparent when a brain scan is performed. This is what we call amyloid PET scan. We also have now biomarkers that can be detected, that is this amyloid or other proteins like tau that Dr. Selkoe mentioned, that could be detected in a spinal fluid and a spinal tap, and that is also being used. And finally, more recently, we now have a test in blood for amyloid as well as tau and other proteins. But for the clinical trials that we're talking about today, the brain scan for amyloid is probably the key component.
Well, thanks to you for the work on these biomarkers, because we know we wouldn't be where we are today without this ability to look at change in the brain and the person living with Alzheimer's. Dr. Selkoe, I want to come back now to talk about the innovation right in front of us, Lacanumab, a size drug in collaboration with Biogen, and the results that have come out And we have phase 3 results in the news. Let's assume these results hold strong in further reports anticipated for this November. Talk to us about Lacanumab. What is it about the results reported for this that shows such promise? What excites you about it? And what are those top line results? Who was studied? And what did you see in the endpoints in this phase 3 trial?
Well, Deborah, I certainly agree that this is perhaps the most exciting therapeutic news in my career. And what we saw on the 27th of September that the company released was a top-line summary that amyloid plaques were lowered, first of all, but that was accompanied by slowing of clinical decline. So the key primary outcome was a test called the CDR sum of boxes, in which patients on Lecanemab had a 27% less decline, a slowing of decline, compared to those who were on placebo in a large trial of almost 1,800 people that was done globally. And so that was the result we've all been hoping for quite a long time. We'll perhaps talk later about the earlier result from Adukanamab, which was also very interesting. But this top-line data looked definitive. We'll be seeing on November 29th the details of that when Lecanemab data are presented at a conference. But the top-line results, which I'm sure will very likely hold up and hold true, suggest that you slow down the Alzheimer process. And in doing that, that happens because you lower the amyloid with Lecanemab, which only has specificity for amyloid. It can't do anything else in your body other than bind and neutralize and remove amyloid. So this in many ways proves the amyloid hypothesis that we mentioned earlier, that if you could get rid of amyloid, there'd be less symptoms, less decline. In addition, the top-line results suggested that 4 other secondary endpoints, which were all checked out in a row, pre-specified, also all hit their endpoints. And those were the degree of amyloid lowering was highly significant versus placebo, and 3 other kinds of tests of cognitive and mental abilities. All 3 of those also were highly statistically significant in favoring Lecanemab over placebo. So this is what we were so long hoping for, a clean group of statistics showing that you can slow the disease. Now, I should mention that the 27% on average decrease in decline on the primary test called CDR sum of boxes, that is an average. So some patients didn't do quite as well as that, but some patients, of course, did better than the 27%. And that scale itself is not necessarily ideal. It's one that many of us in the field choose to test a therapeutic, but it also has scores on it that wouldn't be expected to move in these early Alzheimer patients that we studied in Lecanemab trials. So I would say that this looks like a very impressive result in the top line data, and it may even be better over time than the numbers released on September 27th suggest. And importantly, it looked like the difference between Lecanemab and placebo widened over time as you went through the 18-month trial. So very impressive data. Of course, we need to see the details on November 29th.
So over time, the difference got bigger.
Got bigger, exactly. And
Help us unpack this. I want to throw this back to our panel. And Dr. Selkoe, I know you see patients all the time. If I'm a patient, what difference in my life is it going to mean that I have a 20% improvement in something called CDR sum of the boxes? I mean, what does that mean in my daily life? And I want to ask, if these results, assuming they do hold up, is this clinically meaningful, and to whom? And that term now is being used in so many settings, and it's important to us at Us Against Alzheimer's. So I'd like to hear from each of you, your own thinking about the Lecanemab results and what they tell you as a clinician, as a statistician, as the head of the biggest research enterprise in Alzheimer's in the world, assuming these hold up, is this clinically meaningful for the doctor, and is it meaningful for the patient?
So I'm looking forward to see what my co-panelists think about that question. But for my patients and for the many people, hundreds of people I've seen over the years, it's a 27% on average slowing of decline. And I think many of the patients and families I care for have been asking for just that. They wanted a sense that this is not just galloping away, that there are some levels of breaks. Obviously, we want a slowing at a 50% level. And it's my view that we might get that after the 18-month time point. That is, if we can keep a drug like this going for 30 months or 40 months, there might be a much better chance that we will slow down the disease. But in my view, it's clinically meaningful because anything that goes in the opposite direction of this terrible disease, which is only, as we know, downhill and ultimately is a fatal disease, that is highly meaningful to patients. The term clinical meaningfulness is always difficult to define because it's often doctors who define it, but I would submit that it should be patients and their families who determine that, and we will see, I hope, when this agent begins to be used widely.
The disease is not galloping away. I'll remember that. Dr. Wang, you're a statistician. Is 27% improvement in the CDR sum of the boxes going to be viewed as clinically meaningful?
Again, I'm not a doctor, so I will not use the clinical meaningful, but as a regulator, I would say 27% reduction of the disease progression over 18 months within this population, that is MCI and mild AD, is highly significant. In fact, people don't use or don't argue the 27% or the 25% observed in Adukanamab. Most oftenly, they cited the 0.45 unit or the 0.39 unit as not clinical meaningful because the reference these experts use is the 18 units. That's the full range for the CDR-SB clinical endpoint. From a intuitive perspective, probably most people will think, yes, if the whole range is 18 units, if the drug can only improve the clinical endpoint by less than half unit, that may not sound clinical meaningful or not even close. But the point these people are missing is within this trial, remember, it's only 18 months. And also the population is very mild, MCI patient plus mild population. And within that population and within this duration, even the placebo group, the CDR-SB as a clinical endpoint only deteriorated by 1.6 units, not by 18 units. So that's the maximum margin a drug can improve, 1.6 units, less than 1/10 of the full range of 18 units. So when people use 1.6 as the right reference, then the 0.39 or the 0.45 is not so small anymore. And percentage-wise, it represents either 25% or 27%. Therefore, people should use the right reference to start with, not the entire range, because within 18 months, within this population, that's not the right reference to use. I want to use this opportunity to declare her that because a lot of the experts, even in this field, misuse that 18 points as the reference.
Dr. Wang, I think I actually understand what you're saying, that if you're in the very early stages with MCI and mild AD, you're really holding on to your cognitive capacity. It's subtle, the changes. We know in our work in Us Against Alzheimer's, subtle still means real, that patients in this early stage are truly experiencing change. But you're going to help us, along with our panel, understand how do you decide if a drug is effective in this early part of the disease. Dr. Masliah.
Yeah, thank you. I just wanted to offer also an alternative view or an alternative opinion. I mean, of course, we're extremely excited about these results and looking forward to the more detailed presentation to understand better what they mean. But I would say that it's important to be somewhat cautious on interpreting clinical significance at this point, even though taking into consideration what Dr. Selkoe and Wang have mentioned. But I think what we have learned with anti-amyloid antibodies in previous clinical trials is that they do have the ability to a certain level within 10 to 20% to change cognitive function. I mean, of course, this is the first time where we see a statistically significant number, but we know that, and this is from imaging studies, from the scan studies that I was mentioning before, we know that the accumulation of amyloid in the brain plateaus very early on in the progression of the clinical symptoms of the disease. I mean, the accumulation of the amyloid begins many, many years before in the brain, but then tends to plateau at some point. And the question here is, well, if we decrease the levels of amyloid at a certain stage of the disease, how much would the drug will continue to have a clinical effect if the amyloid plateaus in the brain? And I think that here where we need to take into consideration is that there are other disease mechanisms that come into play. After amyloid have started its process, there are other mechanisms that, I mean, Dr. Selkoe mentioned tau, but there are other mechanisms, for example, the blood vessels in the brain, there is inflammation in the brain. There are other processes that also are at play. So the way we're thinking about this is that, yes, it's very important to hit amyloid. It's very important to decrease amyloid. And this is a very exciting result, but we need to continue working also in targeting other mechanisms downstream of amyloid that in the future, probably combination therapies will become very important. Just to give you an example, we have a clinical trial that is funded by NIA in individuals that have a genetic form of Alzheimer's disease, where they're actually going to be treated with Lacanumab and then with an antibody against tau. And this is the first type of combinatorial clinical study with two monoclonal antibodies, once against amyloid and once against tau. But I think in the future, once we have established amyloid as a target, I think we'll see more and more of these combinatorial studies where multiple targets are hit, and probably there we'll see even a more significant effect. But of course, this is a speculation, but I think it's important to mention that other mechanisms are at play.
Thank you for that. I want to stay with this conversation for a moment and go back to you, maybe Dr. Selkoe. This whole group of monoclonal antibodies, and you just talked about, Eliezer, how you might have combination therapies. Please continue with what Dr. Masliah said, but also come back and help us understand, are each of these MABs, Lecanemab, Gantenerumab, Donanemab, and Aduhelm, are they different, even though they're within the same class? Are we going to expect different results as we see the others read out? Help us understand the class of drugs.
Sure. Well, first of all, I very much agree with what Dr. Masliah said, that we have to move to combination therapies, but to get there, we have to have one first that we can nominate as the beginning point, and that's, it appears, going to be an amyloid-lowering antibody. At least we hope these results will hold up. The four antibodies are not identical. They have somewhat different targets, that is, binding sites on this amyloid beta protein that they bind. And they have been tried out in different ways in different populations. They are early Alzheimer's, that is mild cognitive impairment and mild Alzheimer's, but they're not identical. They're done in different clinical settings, different populations, different countries, and for different lengths of time. For example, Gantenerumab, the next one that we'll report out, is a 2-year period that each patient was given the antibody. So, and by the way, Gantenerumab was given subcutaneously, which is attractive to patients and doctors. because you might not have to come to the hospital or a clinic for intravenous infusion, the way is the case with Aduhelm and with Lecanemab, and actually with Donanemab also. So one of the four is given subcutaneously. Now, we don't yet know whether that will do the trick, whether that will be effective or not. One hopes so. And eventually, Lecanemab will move to subcutaneous also. Eisai has already said that they're starting that process. So the antibodies are not identical. They don't touch or bind the amyloid protein exactly the same way. There's a thought that Lecanemab in particular tends to bind earlier aggregates of amyloid, what we call in the field protofibrils or oligomers, but it also binds amyloid plaque material because it clears it quite dramatically. That was the first secondary endpoint of the Lecanemab trial. So differences, and therefore you wouldn't expect absolutely lockstep findings from the announcement. I'm hopeful that the next two will also show a significant benefit for clinical decline. They will not be curative, just like Lecanemab is not yet curative by any means. But I hope they'll go in the same direction, but I can't tell. I can't anticipate. So one has to respect the fact that this is a class of drugs. If I told you about drugs for high blood pressure, and I said, well, there's calcium channel blockers, and there's angiotensin converting enzymes, and there's diuretics, and they're beta blockers, you wouldn't at this time be surprised that there are different mechanisms. These antibodies are a little closer than that. They work on the same general target, amyloid, but they're not identical, and we don't know that we'll see closely similar results from all four.
Oh, that's so helpful. And I do want to go back what you said a moment ago. It is slowing the rate of decline. It's not reversing the disease. Mm-hmm. Just so we're really clear with our audience today. Yeah. And the meaningful effect, if this is viewed as clinically meaningful, is that the rate of decline, as you said, the disease, it doesn't gallop away. You may be able to put some reins on the horse.
That's right. And so we're hoping for that. Ultimately, if you could use an agent like Lecanemab early on, and that's what's happening, as Dr. Masliah mentioned, NIA and others are supporting trials in a prevention mode before you forget where you parked your car this morning or the name of your 12th grandchild, if you're so blessed. That's when we want to treat in a prevention mode, just like we treat high blood pressure, preventing it before you have a stroke or a heart attack. So in the long run, we want to arrest the disease. I don't know if we're in a position to reverse it. That means stopping all the damage, but more likely preventing it. And that is underway. And Lecanemab happens to be the agent that was chosen because its side effect profile appears to be a little bit better than the other three antibodies.
I want to now move to Lecanemab, and we don't have a lot of time because I want questions from the audience, but I want to our audience the really pressing question: are we going to get access to this drug? Very, very briefly, Dr. Wang, if you were at the FDA or knowing what you do, sitting inside that agency, I'm giving you the crystal ball. Will Lecanemab be approved, and what mechanism or more than one mechanism may be used to approve it, if you think it will be, by the FDA? When might we see the drug? And then we're going to come to access.
I think given the top line result, if it holds up, FDA will absolutely approve it under the regular or full approval, not the accelerated approval. In fact, I would argue even for the Adukanumab, both the clinical pharmacology and the medical division, we both recommended full approval. We did not recommend accelerated approval. Once again, I want to clarify, accelerated approval was eventually chosen by the decision maker as a compromise decision to acknowledge the uncertainty expressed by both the internal and the external experts. So with this kind of convincing data coming from Lecanemab, I'm sure there's no uncertainty, even though there's only one trial, but the size of the trial is almost close to the two independent phase 3 trials. And especially with this very convincing p-value on the primary endpoint, 0.00005, and with all the supportive secondary endpoint also with p-value less than 0.01. I think that's, again, I'm going to use Dr. Billy Dong's words. that is exceptionally convincing. And therefore, I think FDA will prove it with the regular approval pathway.
Well, I can't hear people, but I bet there's some small applause hoping against hope that what you just said bears out. Dr. Selkoe, any response to Dr. Wang's really positive statement?
Well, I certainly don't have the regulatory experience, although I follow the process closely. But if the results on November 29th, when they're presented in detail, are as we see in the top line summary, I would think this will meet the criteria by the FDA for full approval. And so I think we all respect that. And that leads to your next point about access. We know that CMS has made a decision for this entire class of antibodies. I'm on record as saying I don't agree with their choice to make this a class-wide decision. I think they could have decided about Adukanamab and then waited till these Lacanumab data arrive. But in any event, these kinds of phase 3 data in a large trial like Lacanumab should meet the requirements that CMS, the Center for Medicare Services, has for evidence of disease modification. They may ask for some additional evidence by having a registry post-marketing beginning, and that's perfectly appropriate. And we neurologists in the field are certainly going to keep very close track about how this agent is going, whether it's slowing down the process according to what we know about the natural history, and whether the side effect profile is adequate, can be managed. So I'm optimistic that it will get full approval, but I defer to Dr. Wang, who knows more about that, and I'm cautiously optimistic that these kinds of data in the phase 3 Lacanumab study will be helpful to CMS to come to a decision that will allow our patients to access this agent, which they so desperately need, I think.
As I said, this is the question that is at the heart of our work at Us Against Alzheimer's. And I want to, again, unpack this a bit. Do you predict, Dr. Wang, that there will be restrictions on the label, even if the drug is traditionally, if it gets full approval? Will there be restrictions that either come from the FDA, or do you predict, Dr. Selkoe, given what you've seen by the people in charge of Medicare, that there could be restrictions on who might have access to the drug coming out of the starting blocks?
Well, actually, I also want to take this opportunity to give the CMS decision maker some advice. I think they should stop citing a paper that concluded at least 1 to 2 units of CDR-SB should be considered minimally clinically meaningful. I think they should stop citing that paper, especially given we're studying in a very mild population in only 18 months, where the maximum deterioration is only 1.6 units. Therefore, if they still use the 1 to 2 units as the clinical meaningful effect size, they are asking for a cure. This is not the time to expect a cure directly out of this research. They should discuss this with FDA, the expert who understand the trial design and understand how to interpret the data before they make another, in my opinion, improper decision. Because if they follow the same rationale, even the Lacanumab effect size, which is 0.45, is far away from the 1 to 2 units they expected by citing that paper. That's not the right paper to cite, and that's not the right reference to use. I hope this time they can make the right decision.
Dr. Selkoe, do you think there will be a limit on the label as to the people who can be treated with Lecanemab? Do you think CMS will try to limit who can be treated with this drug, assuming it's approved?
Yeah, assuming it's approved, I think the FDA and the sponsor, that's ASI, who directs this process for Lecanemab, will agree to a label that will have restrictions. And there are appropriate use guidelines already made by Jeff Cummings and his colleagues, one of whom was myself, to use Aduhelm in a certain way. With Lacanumab, I think there will be details in the label that this is intended for the population it was tested in, which is very mild and mild Alzheimer's disease, so MCI and mild AD that we call early AD. There will be restrictions. In my view as a clinician, I think it would still be wise to not have patients who are anticoagulants to be offered this agent at this time until we know more. But I think otherwise, the study of Lecanemab was done in a way that it included a spectrum of individuals in the United States that represent the Medicare and the American population. So this should be relevant, these results, to a number of underrepresented minority groups in the United States. So I think there will be restrictions medically. And one of them, of course, is you have to have amyloid underway in your brain, because that defines that you have Alzheimer's. And there will be tests for that. That'll be very important. So it will not be used for anyone who comes to the doctor's office. It will be used for those who have very mild and mild Alzheimer's disease specified by amyloid buildup. And then in terms of CMS, I'd say finally that I agree with Dr. Wang. I hope that they will think very deeply about the totality of scientific evidence. The amyloid hypothesis should not be controversial. In my view, it should not have been near as controversial in years past, but certainly with this Lecanemab result, a lot of that controversy should be removed. And I'm hopeful that while there be restrictions on who can take the agent or who should get the agent, and doctors and patients will have to make that final decision, there will be CMS coverage. That is absolutely crucial. Otherwise, one faces the prospect this is only available to those who can pay the full price and not to the people who are on the Medicare program, which are so many who need it.
I've been looking for questions. I want to open this again to our audience. Please, if you have questions, send them in through the Q&A, but we do have one. It's an important area that, forgive me, we hadn't touched on, and that is, can you speak to us about the safety of these drugs? And is that differentiated across the different monoclonals? And what, should we be concerned?
Maybe I can make a brief comment about that before I close my own remarks. And that is that the safety profiles are generally rather good. The patients don't feel sick, don't notice they have the drug, except for one common side effect, which goes under the name ARIA, or amyloid-related imaging abnormality. It's seen on MRI scans, and it was seen with Lecanemab, but at a lower frequency, a lower prevalence than with the other three antibodies on which we have data so far. So it looks like Lecanemab has an edge right now in how frequent this happens. And it happens about 12.5% in their trial. It happens in placebos a little bit too, this ARIA-E phenomenon that's seen on MRI. But fully 80% of those patients don't know they have the abnormality on MRI. It's not symptomatic, and they feel all right. So the level of symptomatic ARIA-E, or frankly, almost any symptoms, is very low for Lecanemab and somewhat less low for some of the other agents. So I think this can be managed by experienced clinicians. Patients have to be watched carefully. Knowing the ApoE4 genotype is important. I think avoiding anticoagulants in patients who get Lecanemab and these other agents, but there is a way of handling that. So I'm going to pause in my comments at this point.
That was really very helpful and a very important point for us to cover. I'd like to now turn to each of you. The power of us is the theme of this entire summit.
And I'm going to need to leave, as you know.
May go first, because I think you have 4 minutes before you have to leave. If not, can you take 2, yeah, and can you comment?
I'll take just a minute. So your main last question then is? What was
the power of us. What can we in this audience?
I think what we all should do is we should speak to everyone who cares about the future of Alzheimer's disease, that our patients and their families are, quite frankly, appropriately desperate to begin a treatment and to move forward as quickly as possible, since this is a progressive disorder. And so I think speaking to your organization, to all the organizations that care about Alzheimer's patients and their families, lifting your voice and saying, this sounds like good news. We want to understand everything about it. We want transparency. And we're hoping that public agencies will listen to our quest for a useful treatment. Thank you.
Thank you, Dr. Selkoe. I know you have to go. We have 5 or so minutes left with our panel, but we will thank you for joining us and for all you do.
Very happy.
Well, thank you, Dr. Selkoe. Let's
comment.
Yes, please, Dr. Masliah.
Yeah, I think given how wide is your audience and that many of them reached out to patients and families and caregivers, I think another very, very big ask going to the power of us is that, I mean, this is the first time we get a positive result, but it's not the end. There are many other trials that are, as you said, through the pipeline, and many more trials that will come up, some with anti-amyloid, others with combinatorials, others with tau. What does that mean? That we are going to need a lot of public participation on clinical trials. This is something that we've been talking about and trying to make sure that people participate in these studies, both clinical studies, biomarker studies, but also on clinical trials, because we're reaching a point where there's going to be a crunch, a bottleneck. There is so many individuals that might want to participate in a clinical study. So we beg, implore, please participate on clinical studies. It's thanks to the participation of you that we're able to advance science. I mean, you are really the drivers of science. I mean, we put the money, we put the ideas, but it's really ultimately the public out there that makes these things happen. So we're incredibly thankful for those who participated on this study with Lacanumab, but I think we're going to need a lot more participation to advance the field.
Well, how can we help you with that? And how can we get more diverse members of the public in the trials? Just be more specific, because if you had been on this panel 5 years ago, you might have said the same thing. And why is it so difficult in Alzheimer's to get people into trials?
Yeah, I mean, part of the problem was that, especially for recruiting diverse populations, the effort necessary, it's even greater. I mean, these are populations that have not been reached out before. So there is a need for a greater investment, which is in the direction in which we're going right now, have more individuals participating and so on. We also have developed a national plan for recruitment, a strategy for recruitment, utilizing many different avenues and strategies to do this. But definitely investing, in clinical studies in the population and going to the centers, to small groups, to the source, I think, and staying there, maintaining a presence there, it's very critical. And as Dr. Selkoe said, also being very open, be transparent, and staying on point. And I know that you guys also have done a wonderful work on that regard. But
well, thank you for that compliment, because it's a slog. And Dr. Wang, help me here. We've all lived through enough gray hair here to have lived through the AIDS crisis and understanding what ACT UP did. Should we be more vocal? What can the power of this group be with the FDA, and what should it be with CMS? If you could be our band leader.
I absolutely agree that you should be more vocal, because in fact, after last year's approval decision, the majority voice I heard from the media or even from the public, they're all criticizing FDA for making this decision. But when I went to professional meetings like this April to the Washington, D.C. Alzheimer Research Roundtable, then go to the AAIC in August in San Diego, all I heard was positive signals or positive voices. The patients and doctors, they all wanted to use this drug. That's why I was surprised. How come these people's voice were not heard in the media or in any kind of channels? That's why I think you should change this so that the majority of the voice should be heard, especially, the patient's voice. In fact, when FDA made the decision to approve Adukanamab, patient's voice was used as the #1 reason to support the approval decision. And yes, it was a controversial decision, but for us who understand the science, we did not feel this was a controversial decision. In fact, we well predicted the Lacanumab results, even when we were reviewing Adukanumab, because that's when we borrowed evidence from the phase 2 results, from IZI data, from the Eli Lilly data. When we put them together, we felt so confident this class of antibodies, they represent a different class of drug. That's why, when I shared the impressive Lacanumab results with my FDA former colleagues, the response I got was, the p-value was very impressive, but it's not surprising for those who understand the science. So we well predicted the results. So I would say this is not the first time we see a positive result. This is the third time we see a positive result, confirming the previous 2 positive results. Again, we look at both the 301 and 302 results as positive, supporting the effectiveness. That's why our office recommended full approval, even for Adukanumab, predicting the next drug will be also positive, which came out to confirm our prediction.
So how can that patient voice, if it was so helpful, how can it even be louder? You have the opportunity to submit comments to an advisory committee. You have the opportunity to have one person or two, sometimes token, on an advisory committee. And how else can the patient voice this time not let the negative narrative take hold? Any final advice? We have maybe 50, 40 seconds left.
Again, for this one, I don't think there's any need to go to an advisory committee meeting because FDA can confidently make the right decision. But if for any chance there's another AC meeting scheduled for this one, I think all the patients plus the doctors who support this drug should go to the public AC meeting and take advantage of that public speaking period. Everyone should speak loudly about their opinion on both of this drug and Adukanumab, so that all the patients' voice are documented and recorded, so that, well, I think this time the decision will be unanimous, but as a record, your voice should be documented. I know last time in the AC meeting, a lot of patients were already speaking up, but not enough. Clearly not enough to convince the 11 AC members to vote favorably.
Well, I want to say on behalf of our audience that we appreciate the views that each of you bring from your unique positions. And Dr. Masliah, thank you again for leading the Neuroscience Division at the National Institutes on Aging and making sure that this entire pipeline, from basic research to translation to, as you emphasize, the amazingly important investment in new biomarkers, so that you're raising all boats, right? That you're providing tools. And let's hope, George mentioned a vaccine for Alzheimer's. Dr. Masliah, I hope you come back next year and can report that the basic science that will give us a vaccine for Alzheimer's is under development at the NIA. And Dr. Wang, it's often people who've been in the room are not as open with their thoughts, their personal opinions, and this incredibly deep perspective you bring on the regulatory process. Thank you for that. Thank you very much.
Thank you.
So I think now we're going to turn to hearing a pre-recorded video with Dr. Jeff Cummings about the rest of the pipeline. But wait, there's more. So let's turn to that video. and hear from Dr. Cummings before the Lacanumab results were announced. So let's understand that when this was recorded, about the wealth of innovation that in addition to the monoclonal antibodies that we've discussed today. Again, thank you. Let's move to the video. Good morning. We have just heard from our panel about the exciting and complex science behind the anti-amyloid monoclonal antibodies, or MABs. But the MABs are not the only thing in the pipeline. And today we have the rare opportunity to hear from our good friend at USA2, Dr. Jeff Cummings. Dr. Cummings is the Joy Chamber-Grundy Professor of Brain Science, the director of the Chamber-Grundy Center for Transformative Neuroscience at the University of Nevada in Las Vegas. Again, he's a good friend to all of us, but most importantly, a world-renowned expert in Alzheimer's clinical research. Jeff, welcome.
Thank you very much, Deborah. Great to be here.
Would you begin by giving us a sense of the work you have now done over, and I don't even know how many years, to map the pipeline of drugs in Alzheimer's?
Yes, well, this has become a passion of mine, Deborah, as you know. And 8 years ago, I thought, well, it'll be really interesting to look at the pipeline and what's in it. So we did the investigation, and I thought, you know, maybe other people will be interested in this. So let's publish this. And it turned out, of course, people just really like to see what's in the pipeline. And so here's a great thing. It's like a community project. Scientists write to me and say, Jeff, we just entered phase 2. Make sure I'm on your map. So people are really following this and want to get engaged. We publish it every year. We published it for 8 years. It's now funded, importantly, by the National Institute on Aging, and it's called the ACTION, or Alzheimer's Clinical Trial Initiative. And so we're delighted that we now have support for this work. We're going to create a portal so that everybody can have access to this data, probably in 2023.
Oh, that's great to hear. Well, let's not waste any time. Tell us what's in the pipeline. We just heard about the MABs, but what else excites you and should excite us?
Well, Deborah, here is the figure that we usually use when we publish the pipeline. The way it works is that the center circle are the phase 3 compounds, the middle circle, the phase 2 compounds, and the outer circle, phase 1. Green at the top are biologics, mainly monoclonal antibodies. Purple are the small molecule disease-modifying agents. Blue are drugs for symptomatic relief of neuropsychiatric symptoms, and orange are cognitive enhancers. So you can see that there are 143 agents in 172 trials. 31 of the agents are in phase 3. So we've got a lot moving towards phase 3, particularly, as you've emphasized in the panel, the monoclonal antibodies. Phase 2 has 82 agents, mostly disease-modifying, phase 1, 30. The disease-modifying agents comprise 83% of the entire pipeline. So we know that these are the drugs that are being worked on most aggressively. There are cognitive enhancers and neuropsychiatric symptoms. I want to show you one other thing, Deborah, that I think is so exciting. You've asked about what's coming. Well, there are 22 phase 2 drugs that will read out in 2023. We are going to learn so much. So here I've organized them by first, second, third, and fourth quarter. You see that the drugs are roughly equally distributed among the quarters. And what I've called attention to at the bottom is not an exhaustive presentation of all the mechanisms, but just to give you a little sense of the diversity of mechanisms that are being explored in the pipeline now. So you see that we have anti-inflammatory drugs that neutralize TNF, an important target in inflammation. You see that we have PPAR delta gamma dual agonist. You see that we have a glucocorticoid receptor 2 antagonist. You see that we have an A beta aggregation inhibitor and a neuroprotective agent, a sigma 2 agonist. You see that we have anti-inflammatory leukotriene receptor antagonist and a TREM2 monoclonal antibody, really interesting. So my point is here, there's great science going into the pipeline, and we're going to learn so much. Some of these trials will fail, of course. Maybe some of them will succeed and be advanced to phase 3, but we're going to find new drugs as time goes on.
This is just thrilling. We have had a panel talking about the MABs, and we may have already discussed this, but because this is a pre-recorded conversation, when you say disease modifying, what do you mean, Dr. Cummings?
What I mean by disease modifying is targeting a process within Alzheimer's disease that will disrupt the disease process and be seen at the patient level as a slowing of their decline. That's the critical element.
And you just used the words that are close to our hearts at Us Against Alzheimer's. You said at the patient level. We, of course, everything we do, we think of through the lens of the patient and the caregiver. This entire conference for the next 2 days is the power of us. Would you take a moment or two to talk about your perception over decades, you can tell me how long, seeing patients with Alzheimer's, and where today the power rests in the patient and in the caregiver to help accelerate this innovation?
Well, thank you, Deborah. This is so important because it is patients and caregivers who can advance the pipeline. We cannot advance a pipeline without clinical trials. That is the only way the pipeline can be advanced. And the only way we can do clinical trials is with the participation of patients and caregivers. And they are heroic in this, right? We don't know the side effects of these drugs. We don't know the effectiveness of these drugs. So they're taking that risk in order to advance therapy for all patients with Alzheimer's disease. So they have tremendous power. And of course, they also have power as advocates, right? They can demand of the CMS that these drugs be made available to them. They can require that the trials are done with excellence so that their participation is not in any way compromised.
Oh, that's a terrific thought. One last question. We're in a new age where real world evidence is going to be developed to show how these drugs perform in the real world with real patients. Can you leave us one or two ideas of your own about the value of this new pathway? And again, the power of us, the power of the patients and caregivers as we move into developing real world evidence on drugs performances.
Yes, I think we're moving into this great world of real world evidence. The clinical trial is highly artificial. It is a way of determining exactly in a selected population whether the drug affects the progression of the illness. Well, we select patients in such a rigorous way that they do not represent the real world. We exclude patients who have a lot of other illnesses or on a lot of other drugs, or maybe too old or too young to participate in the trial. We need to know how these drugs perform for everyone, for the power of us to be demonstrated in the real world. And I think it's really exciting to see that we're going to affect not just patients in trials. That's only one step in the way of developing therapy for all of us. So I just can't wait to see the real world evidence playing out.
Well, on behalf of everybody in the audience today at the Us Against Alzheimer's Summit, thank you for your time. Thank you for these comments. And most of all, thank you for what you have done and continue to do for people affected by this disease.
Thank you very much, Deborah. Pleasure to be here.
Thank you to Debra and our panel of experts. Coming up at 2:30 Eastern, we'll focus on the importance of brain health and early detection. But not all brains are the same, and the people most impacted by Alzheimer's, Latinos and Black Americans, are the same people struggling with access.